A very good morning, everyone. Can you all hear me? Great. This is my first time to be here and visit this beautiful city, Berlin. So I look forward to our interactions in the next 3 days. So today is my great honor to actually share with you our recent work on how we integrate brain imaging and artificial intelligence with applications in neurological disorders. Throughout our lifespan, we all seek to optimize our cognitive capacity and mental well-being, and we want to maintain our peak function as we age. Brain disorders such as Alzheimer's disease, depression, schizophrenia affect 1 in 3 people at some time of their lives. The global annual cost of all these brain disorders will reach at least $6 trillion by 2030. So as you know, for dementia or neurocognitive disorders specifically, given the aging society worldwide, it will double every 20 years. So this figure needs no introduction. Let's see if I can point. Okay, great. So this figure, which is about the MR phenotype across the lifespan in Nature 2022, needs no introduction. The important part is at the bottom. If you see all the blue box, you can see each of the major neuropsychiatric disorder is associated with a specific age range from early childhood to late adulthood. So in my lab, we focus on 2 vulnerable groups, but today I'm going to focus on the part in the pink box, which is elderly at risk for neurodegenerative or cerebrovascular disorders. In the past 2 decades, as Helen has nicely described, there is amazing advancement in MR imaging. For example, the signal quality of functional MR signals has increased tenfold in the past 20 years. In parallel, as we all are aware, rapid progress in deep learning and generative AI has also begun to impact our lives. So this presents a unique opportunity to understand how human brain works and develop the personalized or precise brain treatments. So in my lab, the long-term goal is to combine multimodal brain imaging and AI to facilitate individualized early detection, prognosis, and proactive intervention. Here in my lab is work centered around the brain network phenotype, which I'm going to introduce later. We have 2 teams in the lab. One is the neuroscience team, centered upon the brain network phenotypes, and the AI team or machine learning team working on brain foundation models, which I'm going to talk about in the second part of my talk. So today, I'm going to show 2 work from the neuroscience part, looking at the brain network phenotype, and then moving on to look at the brain foundation model, how we can use it for brain decoding as well as prediction of future clinical outcomes. So as you are all pretty much familiar with this biomarker model of Alzheimer's disease. So it starts with extracellular amyloid beta plaques, then intracellular neurofibrillary tangles, and followed by brain atrophy. The important part is some of these brain changes can take place years before any symptoms appear. So it's very important for us to understand how we can detect the changes early. So what is brain network phenotype? So this is the structure in my brain of 5 different subtypes of neurocognitive disorders. From the top, you can see Alzheimer's disease have this atrophy pattern in the posterior part of the brain, which is mirrored their memory dysfunction, including the hippocampus, parietal lobe, angular gyrus, and PCC. In contrast, the behavioral variant frontotemporal dementia doesn't have very prominent memory problems, but they do have social-emotional dysfunctions, and is mirrored by this anterior network, including the insular, anterior cingulate cortex, etc. So distinct atrophy pattern can be found in each of the neurodegenerative disorders. So this is a study which took place more than 10 years ago, but it's still very classical. It basically is the first work show that in our living human brain, there is a network-based neurodegeneration. So the cyan color here is the distinct brain atrophy pattern in each of the 5 subtypes of neurocognitive disorders, while the yellow color highlighted how the different regions are functionally connected in healthy brain. And the last row here is how different regions are structurally covariant in healthy brain. So one sentence in summary is the regions that are fired together, they are wired together, and when this disease starts, they also become targeted and deteriorate together. So this seminal work actually raised many interesting questions about selective network vulnerability along the disease progression. So under the AD progression model, the functional MRI as well as FDG PET relates to synaptic dysfunction show changes very early in the disease, even in the preclinical stage. And my group has published a series of work on brain functional network breakdown in neurodegenerative and cerebrovascular disorders. So today I'm going to highlight one important work which I feel like I'm strongly interested in is really the co-occurrence of Alzheimer's disease and cerebrovascular disorder, given it is most prevalent subtype, especially in Asia. We are the first group demonstrating there is a differential brain network phenotype in Alzheimer's disease and cerebrovascular disorder, published in Brain 2017. This is based on not just our dementia cohort from Singapore Memory Clinic, but also we replicated in a Korean cohort, which is the prodromal stage longitudinally, using the objective marker of AD pathology with amyloid PET. So, and then our people basically asked whether the amyloid and cerebrovascular burden have differential effects on the deformal network and executive control network. You can see that the line here in the red color are those people with pure amyloid pathology but no CVD, and they show decline in the deformal network functional connectivity over time. While those people who have pure CVD pathology but no amyloid in their brain, they showed quite the opposite pattern in the executive control network. So we replicate these 2 findings. However, there are problems with these 2, or limitations, I would say, in these 2 work. First is we only focus on specific cerebrovascular lesion, for example, 1-meter hyperintensity or lacunes. But we know that there are multiple cerebrovascular MR markers, like microbleeds, infarcts. So what are the effects of multiple cerebrovascular MR markers on brain functional phenotype? Is it a general effect? Is it a feature-specific effect? And second is how the cerebrovascular-related brain network phenotype interact with AD pathology to influence the future outcomes, in this case, atrophy as well as cognitive decline or dementia severity. So to answer this question, Joanna Chong used our cohort from Singapore and performed this multivariate approach using partial least square analysis based on more than 600 participants. And you can see on the right-hand side, we actually have all the cerebrovascular lesions quantified, the ARWMC, quantification of 1-meter hyperintensity, and then cortical infarcts, lacunes, as well as microbleeds. And on the left-hand side is the general cerebrovascular-related brain network phenotype as marked in the different interaction between the networks. So we found this widespread, non-feature specific effects on brain functional network. And if you narrow down to what are the regions that is important, you can actually see that the cerebrovascular-related brain network phenotype represents a lower cortical specialization and segregation. So basically the diagonal regions is lower and off-diagonal regions is hotter. And you can also see at the bottom part of the regions is a subcortical and cortical connection. You can see an opposing pattern between the cortical and the subcortical region, depending on whether it is related to the sensory motor regions or the higher order associative networks. So this particular pattern is very interesting to us, and we further want to answer how this interact with AD pathology. We use this plasma p-tau1A1, which is a scalable marker of AD amyloid pathology. at that time when it is available. Now, there's an even more accurate biomarker, which is the p-tau217. So using p-tau181 and also the CVD-related brain network phenotype, we get one brain score per participant and at the baseline, and we look at how the two interact in terms of their future neuronal loss up to 5 years, as well as future clinical outcomes in the 5 years. So the specific question is, do they actually have a synergistic effect or additive effect? So the answer is, it do not have any interaction. The two have additive but not synergistic effects on the future atrophy and the cognitive decline. So this work actually addressed an important gap in terms of the AD and cerebrovascular comorbidity related to dementia, and is actually also true in the prodromal stage. In parallel to functional changes, white matter degradation, which is also an early event in AD, and we have done several works using diffusion MRI, focusing on this important novel free water imaging measure derived from diffusion MRI. So just a quick introduction for those who are not familiar with brain free water, is actually rather than treating each tensor and derive all the tissue damage measures, basically they subtract them divide them into tissue compartment and extracellular water compartment, such that we can derive this free water measure, which is indicating higher free water is related to neurovascular inflammation due to possible BBB dysfunction, as well as endothelial damage. Our previous work, as you see at the bottom, have already shown that there are differentiation in the free water between AD with cerebrovascular, as well as vascular dementia. And it's also related to their clinical dementia rating, which is the dementia severity. And one step further, recently we found that given the increasing awareness of the brain and heart connection, as Anna also mentioned, we hypothesized that the higher brain free water, suggesting the cerebrovascular damage, would be related to the cardiovascular dysfunction measured by the circulating blood biomarkers, and that brain free water changes could mediate the relationship between blood cardiovascular dysfunction and cognitive decline. And our data from Singapore suggests that is indeed the case. Let me show you some data. So this is one of the cardiovascular measurements from the blood, and we all know it is related to cognitive function. But the important part of this study is showing that if you look at the brain-free water from both gray matter and white matter, you can see that it mediates, it's a full mediation, association between the cardiovascular marker and longitudinal future cognitive decline. Importantly, if you look at the white matter free water, the mediation is for both the executive function and memory. But if you look at the gray matter free water, you can see this network-specific mediation pattern. The deformal network only mediates the memory, not executive function, while the executive function free water changes only mediate the executive function. So this suggests that the free water changes in the gray matter network regions also follows the brain network phenotype, and most likely is going to be useful for us to, in combination with the cardiovascular blood markers, to identify the high-risk groups for us to intervene early. So Dr. Ji Fang from my group actually performed the free water study in the previous, and now we are very interested in how that may manifest in people with mild behavior impairment. So actually 2 days ago, Lancet just released a paper on the treatment plan for Alzheimer's disease, and with a special focus on how do we treat Alzheimer's disease with behavioral problems. So mild behavior impairment is exactly for that particular question, is how do we identify a person's at risk and faster progression in terms of dementia severity? Is a questionnaire in terms of mild behavioral impairment useful enough? Or actually we should use brain-free water or inflammatory biomarkers like GFAP to actually identify those are truly at high risk. So this is a study which basically we have shown that there are brain-free water differences between those with mild behavioral impairment compared to those without a mild behavior impairment, and their cognitive status, their dementia severity are all comparable. And if you look at various brain measures like cortical thickness, white matter tissue microstructure, and brain free water, we didn't find anything in cortical thickness or white matter tissue microstructure. The only thing we found is higher free water in those with MBI. And as you can see in this graph, those with MBI plus higher brain free water also have faster decline in their cognition, which is in the red lines. Okay, so now you can see the potential of multimodal brain imaging, and now I'm going to change gear to talk about the AI part. So, more than 10 years ago, we have shown that it's possible to predict disease progression from the brain connectome using machine learning. So this is a healthy functional graph at the group level, and we use machine learning to try to link to the disease atrophy patterns and try to understand about disease mechanism of neurodegeneration, for example, transneural spread model. However, this is at the group level, and it's only the first step. Nowadays, we all are very interested in individualized prediction, which is high accuracy, high generalizability to unseen data, stability of the model, interpretability, and sustainability. So the model won't forget if you keep training and training. So all these things motivate us. So my group, in the past 5 years, have made some recent progress on building the brain foundation model based on large-scale neuroimaging data to learn, to decode, and to predict. So today, I will focus on the two work. The first one is how do we use brain foundation model to predict the traits and clinical outcome? And second, how do we use to perform non-invasive brain vision decoding? You may ask me, what is foundation model and why foundation model? So most AI models nowadays in neuroimaging are task-specific. They require large labeled data sets, and they lack the generalizability, so limiting the clinical utility. In contrast, the brain foundation model offers a paradigm shift by enabling the transferable brain representations. The key is self-supervised learning based on large unlabeled data. From the self-supervised learning, through masking and also various other techniques, we are able to learn a transferable brain representations, and then we are able to use it to adapt to multiple tasks, downstream tasks, with minimum labeling and fine-tuning. And this is especially valuable in small clinical data sets and no resource settings. So I'm going to give you two examples, as I mentioned. One is we have developed this brain Japan model, which we are very proud of. And this is the brain dynamics functional MRI data using gradient positioning and spatial temporal masking, which is published in last year's NeurIPS. And then we also have developed approaches is once you have the brain foundation model, how you are going to efficiently adapt them to different tasks. So that is another paper, which I don't have time to go into the details, but if you're interested, you can check this Promptium Brain, the Mikai 2024 paper. So Zhijian Dong is the first author of both papers. Rilin Li is a postdoc, which is joint first author of the Brain Japan. Okay, so let me talk about Brain Japan. So there are a lot of challenges in building brain foundation models. Just to name 3 key questions here, of course there are many other questions and challenges. To me, the first important challenge is how do you locate different brain regions in a transformer-based attention system? Or in short, how do you teach the AI model to know the spatial relationship of different regions, right? It's like the AI model, like a baby, is like walking in a maze. So you need to give them some guidance about the spatial location. And the second question is, does the direct reconstruction of the masked fMRI input make sense? So I want to give you a quick introduction. Is in the self-supervised learning in the old days, everybody just used this mask autoencoder. It's like you mask certain data and use the rest of the data to reconstruct that data. It's like teaching kids, mask a certain letter and guess the missing letter. However, we all know the brain recordings are pretty noisy, and there are a lot of irrelevant signals, like physiology, all these things. So does that work? Our answer is we want to use the JPA model, which I'm going to explain later. The last is how to mask the fMRI input to enable the efficient learning, right? So there are certain principles we could borrow from the neuroscience to guide the learning. Let me walk you through each of the questions and how do we address that in brain JPA model. The first question is location of the regions. We introduced the brain gradient positioning using functional gradients as like functional GPS system of the brain in the attention models. So we propose that using these functional gradients, it is going to be outperforming of the old physical distance or sine cosine, which indeed true. You can see the dark red here, it outperforms all the previous encoding. So we actually give the AI model a brain functional GPS to let the transformer to know the order and the relationship of the different ROIs. So second is, do we do reconstruction in the raw signals or do we do in the latent space? Our answer is we should use the joint embedding predictive architecture, or JPA. This architecture was proposed by Dr. LaCoon and his team in 2023, CVPR. We attend the conference, and then we feel like, wow, this is really a fascinating architecture, and we immediately try to apply to the brain recording, and it works. So the whole idea is that rather than reconstruct the original raw data, we basically transfer that into a latent space and cover the part of the latent space and try to reconstruct that latent. So it works perfectly in our application. And the last question is, how do we learn effectively? So rather than do random masking, right, I cover this, and we basically do a spatial temporal masking. So you can see that we actually divide them into 3 categories. The green one is actually cross-time masking, the blue one is cross-ROI masking, and the red one is double-cross, so different in time and different in space. So by doing this, we are able to teach the AI model to, during the self-supervised learning, to learn the relationship about the interactions between different regions and across time. So let me show you some results about the performance of brain JPA. We trained on 34 million fMRI frames, 70,000 hours of resting state fMRI from UK Biobank data. And then we applied to external data sets like HCP aging, adenine, etc. Of course, the 2 simplest tasks is age and sex. And we compare our model, foundation model, to the previous foundation model. Solver is the second brain foundation model. Brain LM is the first one. And we also compare to state-of-the-art task-specific model using transformer, which is the BNT, Brain GNN, Brain Net CNN. You can see that our performance in age and sex basically outperform all the previous model on the external data sets. And we also try to do mild cognitive impairment and the preclinical AD classification using the adenine database, as also our own Asian database. So across all tasks, it outperforms all previous approaches. And you may ask, so this is just like 5 tasks. You mentioned about the brain foundation model can be applied to a variety of different tasks. So we then go on to actually test it in HCP to let the model to predict the individual traits. In this case, the flanker task performance, measure of the executive function, and mental health traits, which is neuroticism, across our population-level studies. And you can see our model in the red color achieved almost 200% accuracy increase compared to previous method, and for the neuroticism task, and 32% for the flanker. And more importantly is, I mentioned that we only need minimum fine-tuning. So this figure shows that even you don't do fine-tuning, you just take the brain foundation model pre-trained and then use it without any fine-tuning, which means the linear probing. You just add another MLP and just use it. And you can see our model demonstrates a superior linear probing capability. The drop in accuracy compared to fine-tuning to the linear probing is minimum compared to the other model in pink color. As I mentioned, we also want our brain foundation model to be interpretable. We actually analyzed interpretability of our model across different tasks. Just an example here is for the attention patterns for MCI classification. On the left is the adenine database, which is a Caucasian data set. On the right-hand side is a single data set of the dementia clinic. And then the basically different bar represent how different brain networks contribute to the MCI classification. You can see first, the two cohorts give you very similar patterns in terms of the brain network contribution, which is reassuring, right? I mean, if it's very different, I would be very worried. And as I'm doing brain network phenotype in dementia, so I know this makes sense. You can see the highest is the deformal network in the pink color, and then the frontal parietal, the executive function, is in yellow color. And you can also see that in the Asian, the frontal parietal executive control network is more important. It's almost going to beat the deformal network. And why? I can give you the answer because adenine database does not have any person with cerebral vascular disorders. They basically exclude them, okay? But our simple cohorts, we have both AD and cerebral vascular disorders. So, and we know that cerebral vascular disorders is closely related to executive control networks. So that totally explains it. And just like a few months ago, we have developed the brain harmony. So just now, the brain JAPA is solely based on functional MRI signals, and you may wonder how the individual brain anatomy contribute to this model and basically the structural constraints of the functional signals. So the brain harmony is the first multimodal brain foundation model, which basically will align the morphology from structural MRI with the functional dynamics from the fMRI into a unified one-dimensional brain tokens. So by doing this, we can actually outperform the unimodal methods greatly across different various tasks. This has been actually accepted by the neurologists this year, which we're going to present in San Diego in December. That's why I won't go into details, but I already put the paper in the archive. But this is just the first step. Just want to add that we actually include ABCD data in the self-supervised training, so now the model can be extended to even more tasks covering the neurodevelopmental disorder to the aging-related disorders. Okay, so now let me move on to talk about how we can use brain foundation model approach to decode our brain. Here is our mind viz. So basically, the idea is if we put a participant in the scanner and let them to watch movie or look at some images, we take the brain recording data and we put into the AI model, which know nothing about the stimulus, and the AI model can it reconstruct the image and the video from the brain recordings. So using the same concepts, the left-hand side is pre-training, self-supervised learning, unlabeled data. On the right-hand side is using the brain representation learned from the first phase and try to give some guidance to the latent diffusion model, which is a generative AI, right? To regenerate, reconstruct the image and the video. So, give you an example. This is credits goes to Zijiao Chen, who is now doing a postdoc in Stanford. So, if a participant is looking at a giraffe, we record the brain signals. If we use the old traditional approaches, it doesn't work, right? They cannot reconstruct the giraffe. And our approach can reconstruct the giraffe. The semantic accuracy is high. So as you can see, the quantification on the right-hand side, our approach outperformed previous approach in terms of semantic accuracy. But of course, you can see the low-level features in terms of color, the spatial locations still needs more improvement. But just to highlight, this is non-inversive functional memory recordings, so that's why it is more challenging. A few more examples of the image reconstruction. The ground truth is on the first column, ours is the second column, and all other previous methods based on GAN, based on autoencoder, they basically are not so good. And you may wonder, oh, because you're using a latent diffusion model, which is a probabilistic model, is it very stable? So we generate the images like 3 times or even more than that. You can see for each instance of the generation, it has high semantic accuracy and stability. We then extend it to video reconstruction, which provides a more naturalistic setting and mirrors our daily life experience. So here are some videos, the ground truth on the left, the generated one from the AI model on the right-hand side. Again, you can see high semantic similarity between the two, but there are some discrepancy at the low-level features. So we are still improving on it. The important part is that we can also use the approach to identify which region contribute to the reconstruction at each individual and each stimulus level. So that allows us to actually understand the brain and behavior relationships. And lastly, we are also humbled to receive research support from MICAI Challenge Organization Committee. It's like we hope to actually segment some of the very early cerebrovascular lesion, for example, perivascular spaces, and then leveraging on all the expertise we just mentioned. And it's an ongoing work. So with all the technology from neuroscience and AI, we hope that together with everybody in Singapore as well as around the world, and hopefully we can build more collaborations. And then this is one of the studies in Singapore I'm leading as a single study, which is a multi-domain lifestyle intervention study tackled vascular cognitive impairment. So we basically recruit persons at risk for vascular cognitive impairment, 1,200 of them. They undergo 2-year intervention, looking at control of blood pressure, diet, exercise, and cognitive training. And the aim is that we want to use multimodal biomarker, brain, retina, blood, and genetics, together with AI to answer who will benefit from this kind of intervention and why. So we are going to conclude the study by the end of the year. So next time, I hope to report back to you about our results, especially using the brain foundation models. So moving forward, we hope that by integrating multimodal imaging as well as the AI model, we hope to do more work towards the precision neurology and psychiatry. And we need more talent like you guys, and more collaboration across the different continents. And hopefully we can develop personalized and more precise brain treatments to help our patients, as well as have a good brain health plan for the large populations. So with that, I would like to thank the founding agency, all the collaborators, as well as the lab members who contribute to these studies. And very nice picture about Singapore, and welcome all to connect with us and visit Singapore. Just send me an email.